Hello friends, welcome to my channel. Today we'll be studying a topic from general pathology, that is apoptosis, which is a type of cell death. So before starting the topic, let's see what we are going to overview. So today in the first part of apoptosis, we will be studying about the definition, types and mechanism of the apoptosis. Among these topics, mechanism is really important. So now let's start the topic. So since we know that there are two types of cell death, so what's the difference between them? How the cell chooses whether it wants to die through apoptosis or necrosis. We will discuss about these things on today's lecture. So in one word, apoptosis is called as cell suicide. Now a question arises that why a cell wants to commit suicide. So there are two reasons for this. They are, number one reason is that the cell in human body has already performed its function in the body, that is, it is of no more use. Second reason is that the DNA of the cell is damaged to such an extent that it cannot be repaired. So the cell has to do suicide, otherwise it can lead to cancerous cells into neoplastic cells. So before going into the definition, let's see this figure. Let's suppose this is a cell and it wants to commit suicide. So because of any two reasons, that is, either the cell has already performed its function in the body and it is of no use, and other reason is that the DNA of the cell is damaged. Because of these two reasons, the cell wants to commit suicide. And in the cytoplasm of a cell, there is an enzyme known as caspase, which is in inactive form. It is present in all human body in inactive form. And when a cell receives signal that it has to commit suicide, then this caspase will become activated, and after being activated, they will split the entire cell into small apoptotic bodies. These apoptotic body contain a small portion of cytoplasm, cell membrane and nucleus. During this phenomenon, there is no leakage outside, so there is no inflammation, and these are the two hallmarks of apoptosis which differentiate it from necrosis. What are those hallmarks? They are no leakage and no inflammation. But in necrosis, there is bursting, which will lead to leakage and inflammation. Ultimately, a cell known as phagocyte comes and engulfs all these apoptotic bodies, so there is no leakage and no inflammation. Now let's see the definition of apoptosis in the flowchart way. So it is a pathway of cell death that is induced by a tightly regulated intracellular program in which cells destined to die activate enzymes known as caspase, which will degrade the cell own nuclear DNA and cytoplasmic proteins. After that, the cell is phagocytosed and there is no leakage and as well as no inflammation. Apoptosis is the way of elimination of unwanted cells and those cells which are damaged beyond repair capacity of cell. So apoptosis generally involves single cell in contrast to necrosis that usually involve a group of cells. It means apoptosis generally involves single cell, that is, it do not affect its surrounding cells, while in necrosis it leads to or affects surrounding cells near it. Apoptosis is the programmed cell death. It is genetically programmed and it is an energy dependent process. So question arises that central to apoptosis is the utilization of which enzyme? A. Nitrous oxide, B. Adenyl cyclase, C. Caspases, D. CAMP. So what is the answer? Drop down its answer in the comment section. Then after that we will recheck it. Now let's come back to its types. Apoptosis may be of two types. Number 1, physiological. Number 2, pathological. Physiological is programmed cell death, while pathological is unprogrammed cell death, means disease type, and physiological is not disease type. First we will see the physiological causes of apoptosis. Number 1 is embryogenesis. Number 2, elimination of potentially self-reactive lymphocytes. Number 3, hormone-dependent involution of uterus and breast. Number 4, death of cells that have completed their functions. Now let's elaborate about embryogenesis. So let us suppose that this is the uterus where the fetus grows. So during embryogenesis, various organs are formed. For example, how the digits of hands are formed. So let's suppose that this is the hand which is initially in the form of block, where the 5 digits aren't differentiated. Later on it is converted into 5 digits. So how the process is done? It is due to the reason of apoptosis. The cells between the digits undergo apoptosis, that is dying. So by the process of apoptosis, it is converted into 5 digits of hands. So not only in this but also in multiple organ formation, apoptosis is very helpful. Lymphocytes are a type of WBC which kills foreign cells. Sometimes in human body, this WBC becomes abnormal, self-reactive and autoimmune, and they kill their own cells. So they are eliminated from body by the process of apoptosis. Now let's come back to its pathological causes. Number 1 cause is cell death in tumors exposed to chemotherapeutic agents. Number 2, progressive depletion of CD4 plus T cells in the pathogenesis of AIDS. Number 3 is cell death in viral infections. Example, formation of councilman bodies in viral hepatitis, in which hepatocytes undergo apoptosis and form councilman bodies. Now we will see about the mechanism of apoptosis. So, how apoptosis takes place? So there are two mechanisms of apoptosis, that is extrinsic pathway and intrinsic pathway. What do you mean by intrinsic and extrinsic pathway? Now let us suppose that this is a cell, this is the nucleus, and these are the DNA of a cell. Now the cell is destined to die or it has to undergo apoptosis. So if the signal comes from outside that it has to die, it has to commit suicide, then this process is known as extrinsic pathway. It will die through the mechanism of extrinsic pathway. But if the signal comes from inside, of a cell from any organelles. Then this process or this mechanism is known as intrinsic pathway. And finally, the activation of caspase enzyme will take place, and finally the cell will undergo apoptosis. Now let's come back to its mechanism. So apoptosis can be induced through two distinct but convergent pathways. Number one is extrinsic pathway, which is initiated by extracellular stimulus with the help of specific receptors known as death receptors. Number two is the intrinsic pathway. It is the result of increased mitochondrial membrane permeability and release of pro-apoptotic markers like cytochrome C in the cytoplasm. So the mechanism of apoptosis involves two pathways, and those are extrinsic pathway and intrinsic pathway. In each of these pathways there are two phases known as initiation and execution. Initiation phase of both pathways are different. So first we will study the initiation phase of each phases, and then we will go with the execution phase, which is common for both. So let's start with the initiation phase of extrinsic pathway. So let us suppose that this is a cell, normal cell, and this is the nucleus of a cell with DNA inside. And all the cells have receptors present on their surface. These are the receptors known as death receptors. And there are two types of death receptor with same mechanism, and those are FAS CD95 and second one is TNF, that is tumor necrotic factor. So these death receptors are present at a distance from each other. As you can see, a ligand is coming which will bind with the receptor. This ligand will be in yellow color. This is a signal which is in the form of ligand coming from outside. So this is the initiation phase of extrinsic pathway, in which the ligand comes from outside, or signal comes in the form of ligand from outside. So when the ligand binds with the receptor, it will come to know that the cell has to die. So when this ligand binds with the receptor, then the cell will come to know that it has to commit suicide or it has to die. So multiple FAS receptor will come closer. It will come closer and form a domain known as FADD, that is FAS associated death domain. In the cytoplasm, various caspases are present which is in inactive form, and once these FADD are formed, they will activate from procaspase 8 to active caspase 8. Same mechanism goes with TNF death receptor, that is tumor necrotic factor. Here, after when it binds with the death receptor, all the TNF receptor combine and it will also form a domain known as TRAD, that is TNF receptor associated death domain. And finally, the domain formed known as TRAD, it will also convert the procaspase 8, which was in inactive form, to the active form, that is active caspase 8. So in the flow chart here we can see that the Fas protein known as CD95, when it binds with the Fas receptor, that is death receptor of a cell, then multiple Fas proteins will come together and the cytoplasmic death domains combine to form a death domain known as FADD, which is Fas associated death domain, and finally it will activate procaspase 8 to active caspase 8. Also another death domain known as TRAD that will also convert inactive form of procaspase 8 to active form of caspase 8. So this was all about the extrinsic pathway initiation phase, in which it is initiated through specific receptors called death receptors, and those are Fas protein CD95 and TNF receptor. So in this pathway or in this phase of extrinsic pathway, the signal comes in the form of ligand from outside of a cell and it binds with the death receptors, which is in inactive form. After binding, these death receptors of a cell becomes activated and those will combine to form a domain known as FADD or TRADD, that is FAD or TRAD. And finally those domain will convert the inactive form of caspase 8 to active form, that is active caspase 8. So this was all about initiation phase of extrinsic pathway. Now we will see about the initiation phase of intrinsic pathway. So in the initiation phase of intrinsic pathway, the signal comes from inside of the cell. Here you can see a cell with a nucleus and DNA present, and this is the mitochondria of a cell with its inner and outer layer, and between the two layers of the mitochondria there is a protein known as cytochrome C. This is a protein of a mitochondria. So this cytochrome C protein will never come out of the mitochondria to the cytoplasm. Once it will come to the cytoplasm, then it will cause apoptosis. Now, who is preventing this cytochrome C to come out? So there are two guards present in the mitochondria in the gate or transit of a mitochondria. So here you can see that this is the gate, also known as transit of a mitochondria, and these BCL2 and BCLX are known as anti-apoptotic proteins, which are the two guards which prevent cytochrome C to come out to the cytoplasm. And why these guards are known as anti-apoptotic proteins? These are known as anti-apoptotic proteins because these are preventing apoptosis by preventing cytochrome C to come out to the cytoplasm. Till now the DNA was okay, but now the DNA got damaged and it is beyond repair. So the DNA is sending a signal to the mitochondria to kill the cell, that is to commit suicide. Now since the signal is coming from inside of a cell, so it is known as intrinsic pathway. So once the mitochondria receives the signal from DNA, these two guards are replaced by another 3 or 4 guards. known as pro-apoptic proteins, because these will open the doors allowing cytochrome C to come out. Now here you can see that the cytochrome C, on coming out to the cytoplasm, it will activate caspase 9. How it will activate? In the cytoplasm of a cell, the caspase 9 is in the inactive form known as procaspase 9, which is bounded with Apoptosis as a base friend, you can say. So this Apoptosis is keeping caspase in inactive form, but when this cytochrome C comes in the cytoplasm, it will bind with the Apoptosis and take Apoptosis along with it. So now the procaspase will become alone and it will finally activate the procaspase 9 to active caspase 9. So that was all about the initiation phase of intrinsic pathway. So the intrinsic signaling pathways initiate apoptosis involves a diverse array of non-receptors mediated stimuli that produce intracellular signals within the cell and are mitochondrial initiated events. So what we talked in the intrinsic pathway of the initiation phase is that there is the presence of cytochrome C between the two layers of the mitochondria, which is in inactive form. And after the DNA got damaged, DNA sent the signal to the mitochondria that now the cell has to do suicide or it has to die. Then the cytochrome C will come out through the gate or transit into the cytoplasm. After coming into the cytoplasm, it will break the bond of Apoptosis and procaspase 9 and take the Apoptosis along with it, and finally the procaspase 9 will activate caspase 9 into the active form. And this is how the apoptosis process takes place. Now, how DNA damage occurs? It occurs after the exposure to agents like radiations and chemotherapy, that is genotoxic stress. Now let's see the flowchart of initiation phase of intrinsic pathway. So after getting stimulus, the anti-apoptotic molecules or guards known as BCL2 and BCLX are lost, and these are replaced by pro-apoptotic molecules known as BAX, BIM or BART. These will finally increase mitochondrial permeability and release the cytochrome C into the cytoplasm. This cytochrome C will finally activate Apoptosis along with procaspase 9, and this procaspase 9 will finally activate caspase 9. So these are the four anti-apoptotic molecules known as BCL2, BCLX, MCL1 and FLIP. And here we can see the pro-apoptotic molecules that are BAX, BIM, BAT, BAC, P53 gene, Apoptosis and cytochrome C. Now here are a few things which we have to remember. That is, number one is that mitochondria are the most important organelles involved in initiation and regulation of apoptosis. Second one is mitochondrial membrane permeabilization is the hallmark of apoptosis. Now let's come back to the execution phase. So this is the common phase for both extrinsic and intrinsic pathway. That is, it is the convergence point for both of the pathways. So here we can see that the caspase 8 of extrinsic pathway and caspase 9 of intrinsic pathway both will activate another caspase known as caspase 3 and 7, and ultimately this caspase will activate all other caspases and degrade the cell into small fragments known as apoptotic bodies. And finally the phagocyte will come and engulf these apoptotic bodies, and there is no leakage, so there is no inflammation. So here we can see after the activation of caspase 3 and 7 sequentially activates all other caspase. After that, caspase cleaves cytoskeletal and nuclear matrix proteins, and finally cell degenerate and form apoptotic bodies, which will further be engulfed by the phagocyte, and apoptosis process will occur. So here we end about the execution phase. So now here is one question. We have to remember that in the initiation phase caspase 8 and 9 were activated, and in the execution phase caspase 3 and 7 were activated. Now arises a question that how this phagocyte will come to know whether it has to eat a normal cell or a apoptotic cell. How? For this, let's see a diagram. Now let's see a diagram. So this is a normal cell which have a protein known as phosphatidylserine PS. This is present in the inner surface of a cell, of a normal cell, inner surface of a cell membrane, and during apoptosis this PS or phosphatidylserine, this will flip and it will come to the outer surface of a cell membrane. So phagocyte will come to know which cell has PS, those cells should be phagotized or engulfed by it. So PS is a marker for the phagocyte. So the complete process is completed and there is no leakage and no inflammation. So you got it, that how phagocyte will come to know whether I have to eat a normal cell or apoptotic cell. For this protein known as phosphatidylserine is present generally in the inner surface of a normal cell, but during apoptotic cell, this PS will come on the outer surface of a cell membrane. So the phagocyte will see and recognize PS and it will engulf only that apoptotic cell. Now let's see this flow chart. So the phosphatidylserine is a phospholipid present on inner surface membrane normally, but during apoptosis this phosphatidylserine is flipped to outer surface, that is externalization of phosphatidylserine causes its tagging, and finally the phagocyte will see it and engulf it. These alterations permit the early recognition of apoptotic cells by macrophages. And finally the phagocytosis without the release of pro-inflammatory cellular components will take place. The process of apoptotic cells is so efficient that dead cells disappear without leaving a trace and inflammation is virtually absent. And the essential feature is immediate, specific and non-inflammatory nature of phagocytosis. So here we finished about the part 1 of today's lecture that contained definition, type and mechanism of apoptosis. Tomorrow I will upload part 2, which will include the morphological changes in apoptosis, diagnosis of apoptosis and differentiation of apoptosis from necrosis. Till then, please study, share to your friends, like, share and comment your doubts, and also comment the answer of that question in the comment section. Thank you.